Students, welcome to the course on plant embryology. This is very important and basic course of botany in which we will learn about different types of gametes, that is male and female gametes. Then we will discuss about structure and development of different types of gametes. We will learn about fertilization, embryogenesis, gametogenesis, and other phenomena comes under plant embryology. In this particular video, we will focus on the introduction to embryology, what actually embryology is. So, a multicellular organism, it was a single cell, means any multicellular organism, it has been come from a single cell. Initially it was a zygote, and from that zygote in the progressive changes takes place from the early zygote stage, and it will get developed into an entire plant at maturity. Formation of different tissues by the process of differentiation and organization, it leads to an organized plant body. So right from zygotic cell to the organized plant body, there are many changes have been taken place there. It includes differentiation of different types of cells, then organization of different parts including root, stem and leaves. So any plant which is present on the earth's surface, it is not come instantly. It has been come from a zygote by the progressive changes in their life cycle. Different organisms, they can develop differently, means the developmental processes in different organisms, they are different. There is a branch of biology which deals with the different types of processes of various living organisms, or different types of development processes of various organisms. That branch, it is known as developmental biology. This particular concept, it is applicable for all different types of organisms. How they are developed progressively and how their developmental processes are taking place, the branch which studies such type of developmental processes, it is known as developmental biology. Now embryology, it is very basic branch of developmental biology, and it is very closely related to developmental biology. Means when we want to study developmental biology, we should take support of embryology, or when we study embryology, then we have to learn about developmental biology. So embryology, it is the basic branch of developmental biology which deals with the study of structure and development of male and female gametophytes. Means how any pollen grain is formed, how sporogenesis will take place, then how any egg will be formed in respect to female or female gametophyte. Then after, in the embryology, we will study structure and development of different types of gametophytes. After that, we will see the fusion of gametophytes or gametes, then fertilization, how embryos will be formed, how embryogenesis takes place, and finally how seed it will be produced. After that, how seed get germinated, how embryo get germinated, and how entire plant will be developed. Means embryology is the branch of botany which deals with study of a plant right from its seed to up to maturity. So this particular process, it is known as embryology. In angiosperms and gymnosperms, the early stage of development is an embryo, which is a multicellular organized body developed from unicellular zygote. Means any plant we can look now, it was initially a zygote. It is unicellular organism, but in continuous cell division and progressive development, it is being developed into a mature plant. Embryo, through growth and development, develops into a mature plant. So embryo it was the initial part of a plant from which a mature plant has been developed. Now, thus in the life cycle of any plant, two very distinct stages are observed, that is embryonic stage and growth stage. So first of all, embryo is formed and that stage is known as embryonic stage. In this stage, different processes has been taken place and embryonic development takes place into the different stages. Second stage is growth stage. When embryo get developed into a plant, the plant will grow and develop itself. So any plant has two important phenomena in its life cycle, that are embryonic stage and growth stage. The matured plant, it produces gametes with the help of their sexual organs, that is gametogenesis. So after maturation, any plant, it will produce flowers. Flowers contains both male and female gametophyte, that is we know very well. Pollen grains are produced, that is male gametophyte, and in ovules egg is present, and that egg, it is the female gametophyte, and such type of two gametophytes are formed into the mature plant. It is into flower, and this particular process, it is known as gametogenesis. So in the process of fertilization, male and female gametes fuse with each other and produce zygote. Means after maturity, flower will be produced, and in flower different types of gametes are produced by the process of gametogenesis. After maturation of the gametes, both male and female gametes, they will fuse with each other and produce a zygote. So initially both gametes, they are the haploid cells, means with only one chromosome set, but once these two gametes come together, they will form a diploid zygote, and the zygote which will again get developed into an embryo. So zygote, it is unicellular, but once it is get developed, it get divided and redivided to form a multicellular embryo. So when a embryo is synthesized from zygote, the process, it is known as embryogenesis. Thus, the systematic study of all stages from gamete formation to formation of young plant, it is referred to as embryology. So what we have to learn, what we have to study into the embryology, that right from gamete formation, male and female gametophyte formation, to the formation of young plant, all the different stages going on to take place, this particular process, it is known as embryology. In this diagram, we will learn about how different types of embryological processes are takes place. Here you can see this was initially a zygote, it is unicellular, but after its successful division, it get divided into two different cells. Now two forms many, so this will again get produced, and here you can see differentiation. So the cell differentiation is going to take place here, and this is last stage of embryogenesis or embryo development. Now this embryo, it is present into the seed. You can see here, this is embryo. Once it get favorable conditions, that is food from endosperm and water, the embryo get developed, it get germinated. All these are the germinating stages of the embryo, and finally embryo get divided, or it get converted into a mature plant. Here you can see a very mature plant which can be see here with well developed roots, leaves and stem. This plant, here you can see flower, and this flower, it contains gametophytes, male gametophyte and female gametophyte. Here in this first case, you can see this is female gametophyte, it is a ovary. Here you can see stigma, and this part, it is known as anther. Now these anthers, they produce pollen grains, and this particular ovary, it contains ovules. Now ovule contains an egg. So the fertilization will take place by the pollination. So cross and cell pollination will take place. The pollen grains, it will transfer to the stigma, and by the synthesis of pollen tube, here you can see a pollen tube is synthesized, and these are two pollens which are present on the stigma, and through style they will produce a pollen tube. The pollen tube reaches up to the ovule, and it will fuse with the egg cells of the ovule, and after successful fusion of the male and female nucleus, a zygote will be formed. Once fertilization completes, the entire ovary, it will converts into a fruit, and the ovules which are present into the ovary, they get produced into the seed. There are two types of seeds we know, that is monocot seeds and dicot seeds. So both types of seeds are produced by the same process. Now again, these seeds contains endosperms and embryo. Here you can see in the embryonic stages, embryo get developed into the mature plant. So this particular life cycle, it is going on into the life of the plant. Now we will see how and why we have to study embryology, because plant embryology has scope in following various interdisciplinary subjects. Means embryology, it is very important interdisciplinary branch which can be helpful for the study of various other disciplines. Which disciplines are benefited by the study of plant embryology? So here we can see different types of disciplines, that is very crucial disciplines we can say here: plant taxonomy, plant physiology, histochemistry, plant anatomy, plant physiology, organic evolution. So all these are the different branches which are closely related to the embryology, or the knowledge of embryology can be applied into all these branches. Or we can say that if we don't have knowledge of embryology, we can't study all these subjects in details. So they are closely related with each other. Here you can see diagrammatic representation how embryology is connected with other different branches. Embryology, it is connected with plant physiology, taxonomy, systematics, or we may call the plant systematics, biotechnology, histochemistry, organic evolution. So in all these aspects, plant taxonomy, it is very important. Now one by one in detail we will see how it is useful in different fields. Initially we will see the scope of embryology into the plant taxonomy. Embryological data, it serves one of the best source for taxonomy, especially in classification of plants. Means when we are going to classify a plant, we have to take support of embryological data. How? You can see here. First aspect, number of cotyledons in embryo. So in plant classification, when we will focus on angiospermic plants, there are two classes of angiosperms, dicot and monocot. So this particular data, dicotyledons and monocotyledons. So in the initial stage of angiospermic classification, these two classes, that is dicot and monocot, they have taken into consideration the part of embryology. Means if embryo, the seed is dicotyledon or the seed is monocotyledonous. So these aspects are taken into consideration for the classification of plants. So this is very important and most useful data of embryology for the classification of plants. Not only this, forms of ovules also it will applicable in the classification of different types of plants. For example, different plants contain different types of ovules. It may include orthotropous ovule, anatropous ovule, campylotropous ovule. So the plants which are containing these different types of ovules, they are shifted into the particular group of the plant on the basis of the type of ovule. So this data is also very important, and that data is also taken from embryology for the purpose of classification. Then in angiosperm there are two types of ovules, tritegmic and bitegmic ovules. So in angiosperm you can see bitegmic ovules, and in gymnosperm the ovules are tritegmic. So this data is also very important to distinguish angiospermic plants from gymnosperms. What is difference between plants? So this embryological data, it is very important to differentiate angiosperms from gymnosperms. Next point we can take into the consideration, that is nature of female gametophyte. When this is also very important data for differentiation of gymnosperms and angiosperms. If there is embryo sac present, that particular organism or particular plants are known as angiosperm. But embryo sac is without, or the plant are having no embryo sac or without embryo sac, that plants are known as gymnosperms. So these particular gymnosperms don't have embryo sac, but angiosperms, they have well developed embryo sac. So this data is also very important. Then there are different types of fertilization. This is very common type of embryological data which can be used for the differentiation of gymnosperms from angiosperm. So single fertilization, it is the characteristic feature of gymnosperms only, and double fertilization, it is characteristic feature of angiosperms only. So detail about all these we will see in next lectures. Next data we can use in the classification of plants, that is nature of embryo sac. So Oinothera, allium type of embryo sac, and Peonia type of embryo sac. So all these are the types of embryo sacs, and they are characteristic features of various families. For example, Oinothera, that is the Oinothera type of embryo sac, it is characteristic feature of the family Onagraceae. Then allium type of embryo sac, it is the characteristic feature of the family Liliaceae, and Peonia type of embryo sac, it is the characteristic feature of Malpighiaceae family. Now we will see the scope of embryology into the biotechnology. So knowledge of embryology is crucial in organ culture. So when you want to study most advanced branch of botany, that is plant biotechnology. So in that also we must have the knowledge of embryology. So it is very important in the process of, or in the tissue culture practices, because when we are going to undertake any organ culture, means if we are taking any organ, it may be a plant, it may be a leaf, root, stem, or we can take the pollen, then we can take egg cells for the synthesis of new plant in the tissue culture. That particular process, it is known as organ culture. So for this study we must have knowledge of embryology. Plant tissue culture requires knowledge of embryology, that is for isolation of embryo, how we can isolate embryo from different types of seeds. Also some seeds contain damaged embryos, and these embryos naturally cannot germinate. So such embryos can be germinated on the or by the application of tissue culture. So this particular technique, it is known as embryo rescue, and for this purpose also we must have the knowledge of embryology. Then this is very important for isolation of apical meristem, isolation of cambial tissue, and their Next, growth. Embryology helps in understanding the components of the plants, different components of the plant. If you want to understand, we can take support from embryological data. Experimental embryology, that is tissue culture, it helps in standardizing nutrient requirements of organogenesis and embryogenesis. So when we are going to germinate any embryo or any organ artificial, that is on artificial media, then the data from embryology, it is very useful. So the knowledge if we have about experimental embryology, then and then only we can start organogenesis and embryogenesis, that is in artificial conditions. It is essential for the identification of totipotent cells. So the cells which are having ability, potency to develop into a new plant or to an entire plant, that particular concept it is known as totipotency of the cells. So during the process of tissue culture, this particular process it is very important, how we should identify a totipotent cell. If we could not understand totipotent cell, there is wastage of media, wastage of time. So for the identification of totipotent cell, we must have knowledge of embryology. Which cell can germinate, which cell is viable, which is not viable. This particular information we can get from the embryological knowledge. Knowledge of embryology, it can be applied for plant genetic engineering. So this is again most advanced part of biotechnology, and for this purpose also we must have knowledge of embryology. At least basic knowledge we should have. Then embryological data, it is applicable in histochemistry also. So histochemical study of embryonic study provides information about the biochemical substances accumulated during development and differentiation stages of tissue culture. So in the histochemistry we should learn the biochemical substances which are get accumulated during the developmental and differentiation stages of tissue culture. Means these biochemical substances may be reason of developmental and differentiation stages, and due to which we should know the embryonic study or embryo study we must know. Then this particular information is used for determining nutrient requirement of developing embryo. So by this process, if we want to culture or produce or germinate embryo artificially on the media, by this particular knowledge we can get know what type of nutrient we have to supply or provide to the developing embryo artificially. So this data we can collect from the histochemical processes. Then role of various tissues like tapetum, sporogenous tissues, etc. can be better understood by the embryological knowledge. So tapetum, sporogenous tissue, they are the tissues which are present into the different types of gametes, male and female gametes. And what is the role of tapetum? Why tapetum is present in that particular organ? Why sporogenous tissues are there? So this can be better understood by the embryological knowledge. If there is no tapetum, that what will happen? If there are no sporogenous tissue, what will happen? So this particular knowledge we will get from embryology. Means we can get know the role of different types of tissues which are present into the different types of organs of the plant. Now this embryology, it has great scope into the plant anatomy, and embryology and anatomy, they are the very closely related sciences, and they are known as sisters. Means embryology and anatomy goes hand in hand, and they are supposed to be the sister branches of developmental science. Internal structure of an embryo, it provides us information about the position and interrelationship of different regions of an embryo. So If we have the internal knowledge of internal structure of embryo, it will give us an idea about their interrelationship, means interrelationship of different regions of embryo, how they are related to each other. Then nature of tapetal tissue, that is the part of anatomy, developing anther, internal structure of ovule, etc. They can be determined with the help of the embryological and anatomical studies. So both these branches are closely related. To get know the role of any tissue, we should know the embryological data, types of tissue. For that purpose we should know the knowledge of anatomy. So these two processes are goes hand in hand. Now, scope in plant physiology, how this embryology is helpful into the process of or into the plant physiology branch. So, transformation from vegetative to reproductive phase in the life cycle of plant. It happens due to various hormones and growth promoting substances. And this process is well understood with the help of reproductive plant physiology. And this information is very useful in the experimental embryology. So here, for the understanding of plant physiology, we must have the knowledge of embryology. That is, when any vegetative plants enters into the reproductive phase, means it starts flowering. For that purpose, why the plant is get flowered, what are the embryonic characters and what are the physiological characters are important for the process, or what actually happens for the uh, formation of flowers in a plant, what type of condition is required, or what type of physiological changes are going to take place in the plant. This particular knowledge can be get from the study of plant embryology. So in plant physiology, different types of for the understanding of different types of physiological phenomena, we must know the concepts in embryology. Then embryological processes like embryogenesis, sporogenesis, fertilization, organogenesis, fruit and seed development, seed germination, all they are based on the physiological events. So not only embryology is helpful in physiology, also physiology is also useful in embryology. So for better study of embryology, we must take up support of from plant physiology also. Means these two terms are very closely associated. Lastly, we will see the scope of this embryology into organic evolution. So many plant groups are diverse in the nature and developments of embryo. So we know organic evolution, how it will take place. These different groups of plants which are present diversely into the nature, all they are developed from the embryos. By understanding the diversity, one can understand primitive nates and advanced nature of the plants at the embryonic stage. Now, organic evolution means progressively there are some changes taking place into the plants or took place in different types of plants. So, we can understand the primitiveness and advancedness of the different types of plants by the embryonic stages. For example, naked seeds, it is the example of gymnosperms, and closed seeds, it is the example of angiosperm. It is considered that naked seed, it is the character, that is primitive character, and the advanced character, it is the enclosed seeds. So both these plants, they are developed from embryo, but there are some changes in embryonic stages, and these changes lead to the organic evolution. So this particular study, it can be done by the knowledge of embryology. Then Plant belonging to various groups have very less diversity in their characters at adult mature stage, but have distinct diversity in the embryonic characters. Means many plants at maturity they look similar, but when we go to their history, when they were embryos, their character were very distinct and diverse. So this knowledge we can get from embryology. So embryology also helps in tracing the evolutionary lines in plant groups. So this is also very important. So all these are the scopes of plant embryology, how it is associated with the various branches of botany or biology we have discussed here. So this is all about the scope of embryology into the different fields, and this was the introductory video on the plant embryology. In the next lecture we will see the next point which is associated with embryology. Thank you.